Hello, my name is Srinivas and I'm a CDE graduate from Bangalore, India. And I'll be giving a short introduction to my final year project, BGR. So BGR stands for Bacterial Genomic Reconstruction, and the objective of this project is for pre-processing viral metagenomic data or viral DNA samples. So viral data is very important for us to understand because a lot of viruses create a lot of infections and diseases in human beings. But viral DNA is generally plagued with a lot of bacterial DNA, which makes it hard to analyze the data. So before we can talk about the implementation, some insight into how DNA sequencing work will be helpful. So DNA sequencers, or next generation sequencers as they're called now, basically split DNA into substrings. So these substrings are called DNA reads. So reads are generally of the length of hundreds or so. For example, the reads I was using in my project is of length 250. Whereas genomes are generally of a much higher length. So for example, the human genome is 3 billion character long, whereas bacterial genomes are generally in the order of millions and viral genomes are in the order of thousands. Another thing is that the sequences are not perfect. This means they have a probability of error, and to combat this error, what they do is they introduce a coverage factor. So for each base or each character in the DNA set, what it does is so if a coverage factor of n is present, that means that same base will be in at least n separate reads. So this basically has, so that means that some of the reads will have some overlap with each other because these bases will be covered in multiple reads. So generally there are two ways in which we can recover the DNA genome from the DNA reads. So the first way is read alignment. So read alignment is just done by matching each read to a reference genome. So the reference genomes are available due to many projects which have already been done before, like the Human Genome Project or several other projects like that. So in this, you're just using a string matching algorithm and matching each read to a reference genome to see if that particular genome is present. The other way is overlap assembly. So this is used when you want to try to find new genomes. So what overlap assembly is basically, because of the way the reads are sequenced, you can have a prefix and a suffix match. So then what you could say is this read and the other read which have the prefix suffix match are overlapped, and you can basically extend that to get the new genome. So in my approach to recover the bacterial genome, we are using combinations of these two methods. So we don't have all the reference genomes for bacteria, so we cannot possibly use only read alignment. But what we do is that there is a marker gene in bacteria called the 16S rRNA. I don't know what it is, but that marker gene is present in all bacteria. So we use this marker gene to align reads from our data. And so now we know that at least some subset of reads will be mapped to this marker gene. So once we have the subset of reads, which we refer to as the seed, we extend the seed using the prefix suffix overlap. So by combining both these processes, we are able to get all bacterial genomes. So this approach was done in C++, and it was done with the help of two libraries. One is the string graph assembler developed by Jared T. Simpson, and the other is Bowtie, which was used for the alignment phase, which is developed by Ben Langmead. So at the end of this project, I was able to develop a memory-efficient, multi-threaded, and scalable approach in C++, and all my code has been uploaded to GitHub, and you can find it on github.com/Srinivas9804. And yeah, finally, I would just like to thank my supervisor, Dr. Yanni Sun, and her PhD students. Without their help, I couldn't have completed this project. Although my programming skills are good, I basically have zero knowledge in bioinformatics, and they helped me to both understand and complete this project in time.